this last presenter the full opportunity to share her wisdom and presentation. So Kezia, if you could share your screen. I also noticed that you folks are doing really well, and questions are being posed and answered, and so the discussion is going on in behind the curtain, I think was a term I heard a little while ago. And so we're multitasking, and now we're going to hear from Kezia Manlove from Utah State University, and she's going to look more in depth into what drives individual and herd response to MOBI. So Kezia, it's all yours.
Okay, thank you. Can you guys hear me?
Yes, we can hear you and see the presentation.
Okay, fabulous. Yeah, so my talk, I think, will build pretty clearly and directly from Francis's. My role at the moment, as I see it, is to sort of take the highly detailed mechanistic findings that Francis and Tom have put together, and then try and figure out how they scale up to bighorn sheep populations in other contexts. And so let's see if I can advance. There we go. Okay, yeah, sweet. So I've been trying to think about what drives individual responses to die-off events, and there's a whole suite of factors that are relevant here, including substructuring, host genetics, behavior and phenology, the physiology of the host, the pathogen community that's currently present, and then the MOV strain that's circulating. Some of these factors are directly contingent on the environmental context. And I've got several other folks with whom I work will be speaking over the next few days, and they'll deal with some of these factors in a little bit more depth. But for today, I'm going to be focused mostly on genetics, physiology, and the MOV strain type. And so when I came into, trying to think about how these factors would contribute to what we see in the aftermath of a die-off, I was confronted by a bit of a challenge. I wasn't quite sure how to efficiently track what was happening after die-off events, in part because there's a lot of noisy demography out there, and it's hard to identify strong signals associated with particular attributes in demographic data. And so what I'm really going to be focused on today is thinking about whether we might be able to get some kind of a handle on what's happening in these herds by focusing instead on serological data. I'm really interested in these serological data, which are like the antibody test results data that so many of you have collected for so many different jurisdictions. I got very interested in them when I first started working with NDOW a few years ago. I'd just come out of working with Francis and Raina Plowright at the Lustine, and we'd seen some interesting serological signals, but I didn't have a good handle on what the distribution of serological test results looked like. And when when Mike and Perry gave me access to the NDOW data, I was able to kind of look at that for the first time. So what I'm showing you here is just a frequency distribution of the raw numeric percent inhibition results on the Waddell serological test. So you may be very accustomed to thinking about that test as having 3 categories: positive, indeterminate, or negative, but those test results are coupled with a numerical response. And the current definitions that Waddell uses is they call an animal positive if they have a percent inhibition greater than 50%, and indeterminate if they have a percent inhibition greater than 40%. So I looked at the raw data from Nevada, and it just, I fit, there are clearly 2 different peaks in these data. If you just fit a mixture model over both peaks, you get these 2 clear humps in the CLASA data. What's kind of interesting is that if you then look at the probability that a given valued C. ELISA comes from one hump or the other, you can kind of identify how likely a given quantitative result on the serological test is to come from an animal who's truly seen the pathogen before. So I've put my cutoff lines back on here so you can see the 50%, the true positive, that's the bright red line, the 40%, the indeterminate. And then I figured out that the cutoff, where 95% of the time an animal would come from the exposed group, is actually around 25%. So it's quite a bit lower than the cutoff that we currently use based on the Waddle test results. And that's not a negligible number of animals. So I've circled over here for you the animals that fit between about 25% and 40% inhibition on the serological test. And that's in the Nevada data, that's about 140 animals. So it's a lot. And there's a pretty strong implication here. We could potentially be misclassifying some units, or at least some animals, as never having seen MOV when in fact they have seen it. And you know, there are some obvious ramifications of that, but I was really interested kind of in these quantitative data more generally. So I've been trying to think about why some animals show lower percent inhibition than others. And I'm still kind of focused around the genetics of the host, the physiology of the host, and then also the strain type that's involved. And there are kind of three different processes that I thought might be contributing to this variation. The time of when you get your C. ELISA data in contrast to when the animal was actually infected, something specific to the animal or to the strain or the population that you're sampling within, and then just variation among individuals. And the first of these is the time of the measurement. So if we kind of imagine the process that an animal goes through, this is sort of the model that Emily Almberg put forward a number of years ago, where you've got susceptible animals who become exposed, then they become acutely infected, and then eventually recover. We can kind of overlay what we think should be happening with their immune response during that time. So it might kind of look like this, where prior to exposure, they're at some stable pre-exposure antibody level. Then they go through a period of really rapid antibody expansion. Eventually they clear the pathogen and don't get re-exposed, which triggers some antibody decay. And then they drop back down to some stable post-exposure antibody level. So we can kind of map the disease process onto these, what people might refer to as antibody kinetics. But of course, we know from some of Raina Plowright's work and Francis's work that these recovered animals, they don't stay recovered forever. They're constantly challenged by the pathogen. And eventually they might actually switch from being in the susceptible, exposed, acutely infected, recovered category to becoming chronic carriers. And in that case, we might expect their antibody responses to go up and then just stay high because they keep seeing the agent. So this is a super generic model of antibody kinetics. And we know that kinetics can vary among antibodies. We also don't actually know what role the antibody that's targeted by the C. ELISA actually plays in the host immune response. We know it's a good marker for MOV exposure, but we don't actually know what it's doing, at least to the best of my knowledge. So something different could be happening here, but we do have replicated data on antibodies from all over the continent. And so it's a really rich physiological data set, you know, kind of like body condition or, you know, some of the other physiological data sets that we collect consistently. And it might give us some more intuition about what's happening for different herds. So if we wanted to actually infer that curve from real data, we need data where we've got a good sense of when the pathogen came in. We need herds that are relatively well mixed so that we can assume with some confidence that everybody saw the pathogen around the same time. And then we really need longitudinal data where we go back to the herd and ideally to the same animal over and over again and resample them. And this is super preliminary, but we do have data like this for a few herds. So I'm just showing you, as I've aligned all these herds on time of introduction and then plotted out their percent inhibitions. And the curve that's drawn here is actually the fitted line. And I'll just make a note to be really clear about this, that the amount of time that you stay up at the top of that curve is totally conditional on how long you keep seeing the pathogen. So that peak can get spread out if MOV is persisting within the population. So these cases are ones where we know something about when pathogen fade out occurred. But suffice it to say that these temporal dynamics, they have a large effect on antibody kinetics. Animals who haven't seen the pathogen tend to have percent inhibitions that are really low, around 0. Animals that have seen the pathogen recently tend to have percent inhibitions that are really high. And we're only just beginning to learn about how those antibodies might taper off through time after the pathogen is cleared. So then the second feasible explanation for why we might see so much variation that I could actually get my hands around a little bit was that there may just be big variation between different events. So in the in the Nevada data, I broke up the antibody curves according to subspecies. So we've got California's at the top, deserts in the middle, and Rockies at the bottom. And you can see that a lot of animals that are in that space between 20 and 40% are deserts. And there are just many fewer animals there among the Rocky and California subspecies. So it seems like after exposure, the low percent inhibition signal is concentrated in desert bighorns. And in fact, it's concentrated in just a few herds. So I'm showing here animals, the proportion of animals within a herd that fall into that 25 to 40% range. And it's really just a handful of them. And so that's kind of interesting. We've also had an opportunity lately to look at how antibodies increase through time following introduction. And you can see this is 3 different herds here. And I'm looking at a logged timescale. These are big differences in how quickly those antibodies expand within a host. So we're really seeing a large effect of differences between events, between herds or maybe between strains, which is what I was hoping for. I was looking for a way to get some kind of proxy measurement on the virulence of different MOV strains. So then the last factor that I've been able to kind of wrap my mind around so far is that individuals might vary in terms of how efficiently they can mount an antibody response. And I had a chance to look at this in some depth this spring through this translocation event from Taos Pueblo to Antelope Island. So this was a translocation event, or an intended translocation event, where animals were captured and sampled in the wild at Taos Pueblo in New Mexico in November. They were transported to Antelope Island and tested overnight at Wattle. And these animals looked completely healthy, but 6 of them came back either MOV positive on PCR or positive on the ELISA. So Utah Division of Wildlife Resources isolated and eventually euthanized those 6 animals, and we kept the others in captivity. So they went to an elk feeding facility called Hardware Ranch in northern Utah, where my lab and I could monitor them on a regular basis. So we held them in captivity for a period of time, but it quickly became clear that they had become infected with MOV. We began seeing symptoms. We captured them all in the pen about exactly 17 days after the initial capture event. And then they really began to go downhill after that handling event. We had hoped that they would potentially recover to the point where we might be able to release ones that were testing negative early in the spring. But note these dates, this was late March, and everything was shutting down due to COVID right at this time. So we had a couple of mortalities, and then the state decided that they needed to euthanize the animals. And so we euthanized them on March 27th. And this is a really sad event, and not something that I relished being part of it. But on the other hand, we really wanted to get everything that we could out of these animals. And so we have very detailed information on their symptoms through time. Also, through the handling events, we were able to get longitudinal monitoring of their body condition scores, also of their loin thicknesses. They didn't have rump fat that was detectable, so we were measuring loin thickness, which actually was changing over the course of this event. And then lastly, and salient to this conversation, their antibodies. And the one thing that I would draw your attention to here is just how much variation there is among these individuals in how quickly they're mounting that antibody response. There's a weak signal here of age. So younger animals did seem to, or prime-aged animals did seem to mount their responses a little bit faster, but that's right on the edge of statistical reality, if you will. But we did see a really similar, I should say, New Mexico Department of Game and Fish, who were out sampling at the source population, saw similar serological patterns in the source herd, which was really useful to us. So I would say that on the whole, the scale of this individual level variation is relatively small. It definitely individuals vary between one another, but it doesn't seem to be as large as the effect that we see of these different events. But then also, it does seem that these effects are not always tied to environment, because we did see the same sorts of dynamics in the pen as we saw in the field. So that was a little bit reassuring. So, kind of circling back to, trying to understand what does this mean in terms of factors that could be salient here? Well, it certainly seems to me that MOV strain type is tied to the magnitude of the immune response. I'm not sure that that's exactly right. It could be that host genetics is what's really important. Right now, we don't have enough data to separate the MOV strain type from the host genetics. And that certainly seems to be true in explaining some of the variation among these different events. But then I'm fairly optimistic about all the variation that we see within animals, because ideally we would like some quick way or some single sampling event way to determine whether an animal is likely to be a carrier. That would really facilitate the carriage removal experiments that Francis has been working on. And I'm optimistic, given how much variation there was within these individuals in this one event, that there might be some kind of signal there. So kind of in summary, I think that this serological variance is worth considering. It might explain some of the signals that we haven't been able to explain in the past. It also could potentially mean that we are miscategorizing herds or misinterpreting herds as being MOV-free when in fact they have some history of infection. It does pose some interesting questions. So are these weaker immune response events different in some way? And if so, is that because the herds are more resilient, or is it because the MOV strain that's infecting them is just not quite virulent enough to drive a super powerful immune response? Right now, I don't think we have enough replication to separate those two processes. And lastly, or thirdly, I think it's relevant to think about whether we might be able to understand more about host physiology from this percent inhibition. To that end, I've been working with Liz Bowen out of the USGS and UC Davis with some of her gene transcription data, where we have, or NDOW has amassed information on animal disease status, and Liz has put together gene transcripts on those same animals, and we're beginning to try and parse different signatures associated with animals that are acutely infected or recovered or chronically infected. And I think that there's a lot of potential there going forward. But then I think the last thing to be aware of is that there's this variation in the serological responses that individuals induce does introduce some noise into some of our analyses. And there's been a lot of concern about, inconsistencies among patterns in different places. And I think that this might be able to explain some of that variation that's out there. And so I just kind of want to wind up today by saying I think that there's a lot of additional work to be done especially on the host physiology and MOV strain type side. And I'm continuing to work on that, as is my lab. I'm really hopeful that these gene transcription data may help us out quite a bit in trying to understand what's actually happening within the host. On the strain type side, my read of the data at this time, and I spent quite a bit of time looking at it, is that wall-to-wall signals are relatively weak, but the data coming out of case studies is pretty strong to suggest that there are real differences among strains in terms of the demographic responses that they induce. And you'll hear more about that over the next couple of days. But with that, I just want to thank all of the folks who helped out with data collection and who have let me work with their data over the course of this project, as well as my lab, who've put up with all of my all the chaos of junior faculty life with me for a couple of years here. And thank you all for your time. That's all I've got.
Well, that was plenty. That was a lot. Thank you, Kezia, for that. And look at that, it's 12:10, and we've multitasked our Q&A, so we've had our discussion, we've had a wonderful set of presentations. I want to thank all of the presenters that gave us such great information. I don't know about everybody else, but in my head, I can hear a resounding set of virtual applause for all of the presenters and the breadth of the information. I think they've seeded our discussions both throughout for the rest of this symposium, and no doubt we've all got things we can take home and incorporate into our own sheep and goat management programs. At that point, maybe I will turn it back to Kevin. Hurley?
Yes, I'm here.
Here, Kevin?
I'm here, and so, we set 50 minutes for a lunch break, I believe. Something like that. Yeah. And so I wonder if there's even, one or two questions. Otherwise, we can adjourn. But in fairness to the presenters this afternoon, let's start at 1 o'clock promptly. And Susan will be moderating that session. So what do you think, Margot? Is there any, it looks like the Q&A, there's no pending questions, but if somebody has something, otherwise we can break, but let's be back and be ready to start promptly at 1 o'clock.
Yeah, I'd be inclined to that. I think we had a fairly robust Q&A, and that went well. I think I'd rather give people the opportunity to stand up or go and get some lunch or some fresh air and be back for 1 and ready for the next session.
Okay, sharp 1 o'clock start. Thank you all. Thanks, Margot, for keeping them on